Cancer is a growth of cells which is both uncontrolled and in the wrong place in the body.
Cancer is a disease, a cellular disease, a genetic disease of the body cells in which some of the cells grow without the normal controls.
The way we see cancer is an aberration of cell proliferation, of any of the mechanisms that control cell proliferation.
The cancer is the human disease, and that is the disease where the cells are proliferating very rapidly, and sometimes the cells are metastasizing to the another places of the organ, and finally they will kill the human beings.
Yeah.
In Japan, they call it Gann, but it's the same problem we face the world over, cancer. This patient has cancer of the large intestine. At this hospital in Tokyo, doctors are performing a biopsy, locating the site of the tumor and removing some tissue for analysis. They'll find out more about how serious the problem is and how far the cancer has spread. But they will be no nearer a cure for this patient. His chances of survival beyond the next 5 years are less than half. Cancer accounts for 1/5 of deaths in the developed world, and yet surprisingly little is known about how and why it occurs. It's now clear that cancer is in some sense a genetic disease. For every cancer, it's thought there is an oncogene, a particular gene involved in the formation and spread of tumors. But at the same time, the environment has a major role to play. Carcinogens are all around us, in the air we breathe and the food we eat. For geneticists trying to understand the patterns of cancer in the population, these are all relevant factors. But for the people of Japan, an event in 1945 offered them and the world a unique insight into carcinogenesis. The atomic bomb, which dropped on Hiroshima in August 1945, killed 64,000 people through heat, blast damage and radiation. Apart from the terrible legacy of death and destruction left by the bomb, there were many who survived in the city, and they became the subjects of intense analysis by life scientists. It seemed as though the ultimate weapons of destruction were going to yield valuable scientific insights into life itself. One of the main areas of study has been the influence of ionizing radiation on the genome. It has been extensively studied by geneticist Dr. Akio Awa.
The importance of the effects of ionizing radiation from atomic bomb explosion is to what extent the ionizing radiation would cause genetic damage in man.
In 1950, there was a census of the 110,000 bomb survivors of Hiroshima. This formed the basis of a unique genetic archive here at the Radiation Effects Research Laboratory overlooking the city. These files contain detailed records of all atomic bomb survivors in Japan. Not just medical histories, but also where they were when the bomb was dropped, how much shelter they had from the blast, and a calculation of what radiation dosage they received. There are also files and data for their children. The files are currently being put onto computer. But whatever form they end up in, they should enable researchers to assess the real genetic effects of ionizing radiation. But the work at RERF is not just about checking records. Besides their research work into the effects of the atomic bomb, they run a clinic which enables them to continue to investigate the consequences of the bomb 40 years later. In the clinic, they continue to monitor the health of the population of the city of Hiroshima as part of their work. This woman was 36 at the time of the bomb. She comes to the clinic here twice a year for an extensive medical examination which contributes to the millions of facts kept on bomb survivors. The examination will include everything from blood pressure and body scans to interviews about their general health. It's all part of the elaborate system of healthcare the Japanese have throughout the country. Geneticists have now been studying both the survivors and their children for 40 years. What conclusions have they reached? Dr. Awa of the Radiation Effects Research Foundation.
And recently we initiated chromosomal screening that since chromosomes are known to be very sensitive to ionizing radiations, and if the chromosomes in the pentagerm cells is affected, we would expect to find more children carrying chromosome abnormalities among the children born to the bomb survivors as compared with the control children. And another thing we have done in the recent past is also screen the protein variants, which are the endpoint from the gene mutations. And none of these studies were able to demonstrate the difference between exposed people and controls. So that our conclusion so far is that we simply cannot demonstrate any genetic effects among the children born to A-bomb survivors.
But one major effect of the bomb was to increase the incidence of cancer in Hiroshima. Overall, there was a 5% increase of cancers amongst the survivors, but leukemias, cancers of the blood, increased by over 60%. For this reason, a lot of research is still going on at the Radiation Effects Research Foundation into abnormalities in the blood of bomb survivors. This sample of blood is being analysed by a cytofluorescence machine. Individual proteins in the blood have already been chemically stained. By shining laser light onto the blood, the machine automatically counts the number of blood cells containing the stained proteins. Proteins are produced by the transcription of DNA, the genetic material. So, by using sensitive techniques like this, it is possible to deduce how much genetic alteration has been caused by ionizing radiation from the bomb. Many chromosomal abnormalities have been found in survivors' blood, but perhaps the most significant difference is found in T-cell lymphocytes. Dr. Akiyama and his radiobiology team have been studying the T-cell response in detail. The T-cells are part of the body's immune response system, generating antibodies in response to foreign antigens in the blood. If the response is plotted against age, it falls off like this in a non-exposed population, part of the normal process of aging, but like this in the population exposed to radiation. There is a significant difference, and it might give a clue as to the origins of leukemias and other cancers.
We are now collecting a tissue from surgical specimen from airborne survivors when they are receiving operation of cancer in order to detect oncogene activation by the radiation and also amplified oncogene expression on the tissue that will be detected by using a monoclonal antibody against several oncogene products.
Although the Hiroshima bomb has led to a statistical increase in cancer, it is still not clear why people get cancer. Studies of cancer today focus both on the environment and on oncogenes, the genes implicated in the initiation and spread or metastasis of cancerous cells. But all cancers have one feature in common: the permanent alteration of cells.
How can you permanently alter a cell? We know of only two ways. One of those ways is by altering DNA. Since DNA is the fundamental blueprint of the cell, you alter that, the cell's altered. And for it to go back to normal, you'd have to reverse that process of change in DNA, and the probability of that is so low that even if it happened in one cell, there would already be so many other cells that had that change that you'd never even know that you'd seen the reversion. So that's irreversible. But we know of another process in cells which is equivalently irreversible, and that is differentiation. The difference between a skin cell and an eye cell or a hair cell or a liver cell is a very permanent change in cells. It is so permanent that if we take cells and put them in culture and grow them in the total absence of the rest of the body, in the total absence of any sort of particular design stimulus for 50 years, or I guess experimentally for 20 years, we've done the experiment. And then you go back and ask, does that cell have the hallmarks of the kind of tissue from which it was taken? The answer is yes, it does. And you can show very easily that that cell knows exactly what kind of cell it was supposed to be.
All cells contain a set of genes which code for proteins involved in normal cell growth and differentiation. These so-called proto-oncogenes can be activated into oncogenes, whose protein products are either present at an abnormal level or are modified in some way, so transforming the cell into a cancer cell. But how can an oncogene arise from a normal cellular proto-oncogene? When a cell is exposed to radiation or a chemical carcinogen, point mutations can arise in the proto-oncogene. The protein this proto-oncogene codes for is altered and may induce one or more changes leading to cancerous growth. And once altered, the cell almost never reverts to normal.
The hallmark of cancer is stability. Once a cancer cell is a cancer cell, it never goes back to being a normal cell. And so whatever alterations are involved in making a cell a cancer cell have to be basically permanent alterations of the cell.
Human cancer is very difficult to study by the basic scientist because it's a very, very final product. By the time human cancer causes any pathological disturbances, it is too late for the molecular biologist to go in and to examine what's going on.
The cancer story is made more complex by the fact that there are also several mechanisms for carcinogenesis. Nobel Prize winner Dr. Howard Temin.
It wasn't until 1970 when in my laboratory and David Baltimore's, the enzyme in the virus particles of retroviruses, reverse transcriptase, was discovered, and soon thereafter infectious DNA of retroviruses was discovered, that it was accepted universally that reverse transcription existed, and this was the way retroviruses could cause cancer, because a gene in the retrovirus could be added to the cell DNA.
A retrovirus infects a cell by injecting its RNA into the cell, leaving its outer protein coat behind. The RNA of the virus is copied into double-stranded DNA by the enzyme reverse transcriptase, which is supplied by the virus. The viral DNA then becomes integrated into the chromosome of the host cell. When the host cell's DNA is transcribed, the viral DNA is also transcribed into RNA. Most of the viral RNA contains information for making new virus particles. These new viruses will form new protein coats and leave the cell to invade new host cells. But the viral RNA also contains information coding for proteins involved in converting the cell to cancerous growth. This portion of the gene is the oncogene.
The first human cancer genes turn out to be the same genes previously identified in 4 strains of acute transforming retroviruses. And these genes are what we now known as RAS genes.
So, RAS is a human proto-oncogene, as well as being found in carcinogenic retroviruses. Although these retroviruses are not the main cause of human cancers, they can offer important insights into how cancer occurs. This focus assay is being done in Howard Temin's laboratory in Wisconsin. First, a uniform growth of cells is achieved in many different dishes, and the growth medium removed. The cells are then infected with the virus to be tested. Will it turn the cells cancerous? This is what the assay, or test, will find out. The host cells will contain proto-oncogenes such as RAS, which will be altered by a carcinogen and so turn the cell cancerous. The carcinogen may be a chemical compound, radiation, or, as in this assay, a retrovirus. After a week's incubation, the cells are ready for examination under the microscope. The transformed cells create a focus of altered cells which show up clearly as dark regions amongst the untransformed cells. The number of foci is proportional to the number of infectious units of the virus added to the cell culture. The work is slow and difficult, but for molecular biologists there are other difficulties too. Oncogenes are not a tidy group of genes with clear functions, but may be very diverse.
Oncogenes have been found in essentially each one of the steps that goes from the outside of the cell to the nucleus of the cell. For instance, some oncogenes have been found to be growth factors. Other oncogenes have been found to be the receptors of those growth factors, the molecules that are inside the membrane, half outside, half inside. Other oncogenes, such as the RAS oncogenes that we were referring to before, are proteins that are able to transduce the signal from the growth factor receptors to the inner side of the cellular membrane. Then they have been oncogenes have been identified as cytoplasmic oncogenes. And then finally, the last group would be the ones that have been found in the nucleus. So essentially, you have oncogenes throughout the entire process of signal transduction from the outside of the cell to the nucleus of the cell.
But there's a huge core missing in this information, and that is the mechanisms inside the cell that allow this gene to be a cancer-causing gene. What happens from the moment when the gene is modified to its effects on the cellular metabolism? And there, the huge lack that we have is of our understanding of what controls the growth of cells, what controls the migration of cells in the body, what controls where cells settle down and where they're able to grow and where they're not able to grow. And all of those are really forefront areas of research.
New computer technology now makes it possible to photograph and analyze in detail cancerous growth in the human body. Endoscopy and surgery now can help more than ever before in the fight to control or remove cancer when it's found. A single altered cell can lead to a tumor like this, which can spread and eventually kill the host. But cancer is a genetic disease, and cancer genes are essential constituents of the cell. One surprising fact about cancer is that different patterns of the disease are found in different countries. The three most common forms of the disease are cancer of the lung, the large intestine, and the breast. If you look at the incidence of lung cancer in different parts of the world, Japan has one of the lowest rates of incidence. The rates in America and Britain are both considerably higher. Much the same is true of female cancer of the breast in these three countries. It's when you look at stomach cancer that a different pattern is found. Japan, America, and Britain. It's different patterns like these which might reveal more about carcinogens.
We are thinking that the lifestyle, especially the dietary habit, are playing some role in the differences of these patterns of the cancers between Japan and European countries. For example, in Japan, we are taking a lot of the salt, and instead the people in the Western countries are taking a lot of fat and also the high proteins and so on. So this high fat diet and also the high protein diet are claimed to increase the incidence of the colon cancers, but the diet which got the very high amount of the salt is claimed to cause the very high frequency of the stomach cancers. So these differences, especially of the dietary habit, are much involved in the differences of the patterns of the cancers between Japan and European countries, I guess.
There is as yet no known mechanism by which dietary habits can lead to cancer, although carcinogens are abundant in the environment. Statistical links with cancer have been found in overcooked meat, in coffee, tobacco, alcohol, and in a variety of food additives. Any of these in sufficient quantity is implicated in carcinogenesis. The role of diet in cancer patterns is highlighted in research into Japanese immigrants to California. Stomach cancer in native Japanese is reduced in first-generation immigrants to America and decreases still further in their children. So diet is an important part of the environment as a carcinogen. In 1983, the British Imperial Cancer Research Fund published a list of factors which contributed to the development of cancer. Diet was at the top of the list. Ionizing radiation in the environment comes a long way further down. So perhaps in the short term, the key to reducing the incidence of cancer lies in trying to change people's habits rather than in genetic or medical research. In Japan, there is a major screening program for the early detection of cancer. For example, it's routinely done at the RERF as part of their general health clinic work. So far, such screening is the only possible step that can be taken towards the eventual elimination of cancer. Japan now has a major 10-year program, which is looking at public health measures which will reduce cancer incidence in society. Only in this way do the Japanese see any hope of conquering the cancer problem.
It's very hard to evaluate, but according to some estimation, probably the 1/3 of the decrease in number of the stomach cancer in Japan could be because of the mass screening activities.
Through the Foundation for Promotion of Cancer Research, we are now distributing this kind of the calendar for the public. And there it says there are 12 recommendations for cancer prevention. In this calendar, they are written in Japanese, but they are saying that have the nutritionally balanced diet and have a variety of types of food and avoid excessive calories, especially as fat and so on. These kind of the recommendations are made by the Ministry of Health and Welfare. So this is the typical support by the Japanese government for the cancer researches and also the practical application of the cancer research to the public, I guess. So this kind of the recommendations are getting, well, effective, I really hope. And within 10 years or 20 years, maybe we can have the 10% or 20% decrease of the cancer incidence in Japan.
Files of the bomb survivors in Hiroshima, where leukemia and other cancers are well documented, may yet provide some of the answers to the continuing mystery of what causes cancer. And no doubt new techniques of genetic analysis will help. When that time comes, scientists will have tissue samples to work on. These samples from the bomb survivors are being stored in liquid nitrogen until the science of genetics is refined enough to analyze them. Until then, solutions to the riddle of cancer seem unlikely to be found.
I think we have, in fact, the genetic set of answers more or less staring us in the face, and to a large extent it's a lot of work now to put it all into place, to find out in a particular kind of human cancer which oncogenes are activated, and that's beginning to happen.